Welcome, budding biologists, to your first biology screencast. In this screencast, we'll start Chapter 1 and talk about the process of science. At the beginning of each chapter in your packet, you'll find driving questions. Use the driving questions to preview the material and to focus your learning and attention as you watch the screencast. In the future, I'll leave these driving questions out of the screencast. In this chapter, we'll look at the process of science, the scientific method, and how scientists do their work. Here's a photo of two marine biologists studying coral reefs and collecting data underwater. Each chapter starts off with a real-life case study that helps us understand the topic a little bit better. This chapter's case study revolves around caffeine. You may have seen headlines in the news that say things like coffee causes cancer, coffee prevents some diseases, or caffeine can be poisonous. How does science produce these headlines? How do we as the public evaluate these headlines? How do we prevent ourselves from being swayed by misinformation? Caffeine is a common ingredient in many of the beverages we consume. A can of Coca-Cola, for example, contains 34 milligrams of caffeine, and a cup of coffee contains 108 milligrams. Let's start off by asking ourselves, what is science? Science comes from the Latin word for knowledge, and science is a method that we use to answer questions about the natural world. It's a process of using observations and experiments to draw evidence-based conclusions. All in all, it's a way of knowing how and why things have happened in the past, present, and future. When scientists do science, the goal is to draw conclusions. Scientists will gather and examine the best scientific evidence available. They'll ask themselves, was the science performed properly before they'll accept any conclusion? Conclusions are never set in stone. They can be modified in the future with new information. Science is a never-ending process. It's ever evolving, and we're always learning more and more. Your textbook uses infographics to further explain many topics and ideas. For example, this infographic from the textbook shows how scientific headlines, specifically about caffeine, may reduce or simplify the science behind them. Science starts with observation. Scientists will begin with interesting, informal observations and anecdotal evidence. Oftentimes, these observations and anecdotes are unreliable and untested. When we refer to anecdotal evidence, we're talking about personal stories about specific incidents and experiences. For example, someone may tell you that caffeine gives them headaches, and as a scientist, you might test to see if that's actually true. The next step is to formulate a question. We can take our observations and anecdotes we've collected and devise a question we can test scientifically. In our caffeine example, we can ask, does coffee help improve mental performance? After formulating a question, the next step is to study previous research. Scientists do not want to reinvent the wheel. They'll read relevant literature, they'll see current information on the subject of interest, and they'll study peer-reviewed scientific literature. When we refer to peer review, we're talking about the review of scientific work or scientific articles by experts prior to publication. This makes sure that the authors of the work have appropriately designed and interpreted their research. It also weeds out sloppy research. This increases the overall credibility and trust in science. After you look at previous research, you'll use that information to formulate a scientific hypothesis. A scientific hypothesis is more than just an educated guess. It's a testable and falsifiable explanation for a scientific observation or question. When we say testable, it means that we can design an experiment or non-experimental study that will either support or reject the hypothesis. When we say falsifiable, it means that we can collect data that might show that the hypothesis is wrong. In our caffeine example, we might have a hypothesis that states caffeinated coffee improves memory. This diagram shows that it's possible to produce multiple hypotheses as long as they're all testable and falsifiable. Many explanations are not scientific hypotheses. These explanations cannot be tested or refuted, so they fall outside the realm of scientific explanation. Examples include ethical explanations of right and wrong, or religious explanations that are difficult to test or falsify. One thing we need to understand is that a hypothesis is never fully proven true. If false, it is rejected and no longer considered a possible explanation. If the data do support the hypothesis, it is accepted until further testing and data show otherwise. In science, we must change our ideas if they're not supported by evidence. After you formulate your hypothesis, you'll test it using experimentation. An experiment is a carefully designed test of a hypothesis. The results of an experiment will either support or rule out a hypothesis. Let's look at an example of an experiment where we measure the effects of coffee drinking on a group of participants. In an experiment, you'll have two groups. The first group is called the experimental group. This is the group that will experience the experimental intervention or manipulation. We can also say that they experience the independent variable. The independent variable is the factor being deliberately changed in the experimental group. In this case, it's caffeinated coffee. This infographic from the textbook shows how we might set up this experiment, with the first group called the experimental group and the second group called the control group that we'll talk about next. The other group is called the control group, and this is the group that experiences no experimental intervention or manipulation. We use the control group as a basis for comparison. This is the group that we do nothing to see if doing something had some effect. Our experimental group will receive a placebo, which in this case is decaffeinated coffee. A placebo is a fake treatment that resembles or mimics the experience of the experimental group. As the experiment proceeds, we'll measure a dependent variable and look for results. We'll analyze the dependent variable in both the experimental and control groups. In our example, the dependent variable is mental performance. We might look at performance on a memory test in both the experimental and control groups. The observations and data we collect during the experiment will make up our results. We can then use the results to draw evidence-based conclusions.